Our opponent's first claim was that embryonic stem cells are not human beings. But like my partner previously stated, human embryonic stem cells are human beings. According to Sneed O'Connor, who is the associate professor at Notre Dame Law School, he said that the relevant science confirms that 5 to 6 day old human embryos used and destroyed in stem cell research is a complete, living, self-directing, integrated, whole individual member of the human species, who, given the proper environment, will, if all goes well, move itself along the trajectory of human biological development from the embryo to fetus, to neonate, child, to adolescent, and to adult. President Bush made it clear that his federal funding policy was driven by two premises. First, that embryos are living members of the human species. And second, that all such members of the human family are entitled to equal moral respect, sufficient to preclude that their unconsented use and destruction in biomedical research. The first premise is confirmed by modern embryology, and the second is grounded in the classical liberal principle of equality, reflected in our nation's foundational legal instruments. According to Robert P. George, who's a McCormick professor of jurisprudence at Princeton University, said that the adult that is you, the same human being who, at an earlier stage of your life, was an adolescent, and before that, a child, an infant, a fetus, and an embryo, even in the embryonic stage, you were a whole living member of the species Homo sapiens. You are then, as you are now, a distinct and complete, though of course, immature human organism. Our opponent's second claim was that embryonic stem cells have multiple benefits for patients and treatments for potential problems for patients. Well, like my partner previously stated, embryonic stem cells are flawed. According to Richard Hayes, who's the executive director of Berkeley, California-based Center for Genetics and Society, says that a major hurdle to the therapeutic use of embryonic stem cells extracted from in vitro fertilization embryos is the expected immune rejection problem. Cells or tissues obtained from in vitro fertilization embryos would likely be identified by a patient's immune system as foreign and thus rejected. Embryonic stem cells are also prone to trigger the development of tumors, usually benign, known as teratomas. And according to Maureen L. Condict, who is the associate professor of neurobiology and anatomy at the University of Utah, so that more than a dozen papers over the past 5 years, 5 years within the past year alone, have shown tumor formation in animals treated with differentiated embryonic stem cells derivatives. In several of these studies, a shocking 70 to 100% of the experimental animals succumb to fatal tumors. And according to Bernadine Healy, who is the former head of the National Institute of Health, wrote about a report from Israel published in PLOS Medicine in late February that shows embryonic stem cells injected into patients can cause disabling, if not deadly, tumors. The report describes a young boy with fatal neuromuscular disease called ataxia telangiectasia, was treated with embryonic stem cells. Within 4 years, he developed headaches and was found to have multiple tumors in his brain and spinal cord that genetically matched the female embryos used in his therapy. His experience environment of a fast-growing embryo, after all. Though they are tamed in petri dish to be disciplined, mature cells, research in animals has shown repeatedly that sometimes the injected cells can run wildly out of control, dashing hopes of tiny human embryos benignly spitting off stem cells to save grown-ups without risk or concern. According to Maureen L. Condict, over the past 30 years, hundreds of billions of dollars and countless hours of research by dedicated professionals worldwide have been devoted to solving the problems of immune rejection and tumor formation. Yet these issues remain serious scientific and medical challenges. Our opponent's third claim was that embryonic stem cells are the best possible choice when it comes to treatment. Again, like my partner stated, embryonic stem cells should be abandoned in favor of alternatives. According to the Domestic Policy Council, scientists have found adult stem cells in many more tissues than they once thought likely, including the brain, bone marrow, blood vessels, skeletal muscles, skin, liver, and other body parts. Scientists have also reported that adult stem cells may in some cases exhibit the ability to form specialized cell types of other tissues, a characteristic known as transdifferentiation or plasticity. Some experiments have even suggested that certain adult stem cell types may be pluripotent. Adult stem cells have some important scientific advantages as potential therapies. For one, since adult stem cells can be taken from the patient's own tissue, they are genetically matched and would not be rejected by that patient's own immune system. As of April 2, 2007, there have been 1,373 publicly available clinical trials related to adult stem cells, including 671 that are currently recruiting patients. Over the last couple of years, clinical trials using adult stem cells have produced encouraging improvements in patients suffering a range of disease and disorders, including leukemia, lymphoma, diabetes, advanced kidney cancer, and several inherited blood disorders. In May of 2005, the President's Council on Bioethics issued a white paper reviewing possible non-destructive sources for additional stem cell lines in the future. Alternative sources of human pluripotent stem cells examined the ethical, scientific, and practical prospects for a range of different approaches and recommended specific investigations that could advance these techniques. The goal of these alternative approaches is to produce the functional equivalent of embryonic stem cells without harming embryos. One of the most promising possibilities is the adult stem cell reprogramming. Recent work by two research teams, Kevin Egan and Chad Cowan in Harvard Stem Cell Institute and Shina Yamanaka Katsutoshi Takahashi at Japan's Institute for Frontier Medical, suggest that it may be possible to use chemical and genetic factors to reprogram an adult cell to function like an embryonic stem cell. This approach could prove capable of creating cell lines for the study and treatment of disease, without many of the ethical dilemmas associated with the creation and destruction of embryos. In January 2007, landmark research from Wake Forest University has suggested yet another alternative. Amniotic fluid investigators discovered contains highly flexible stem cells shed by the fetus. These appear to have all the valuable qualities of embryonic stem cells, plus some advantages of their own, like greater ease and speed of culture and no tendency to produce tumors. Meanwhile, they are comparatively easy to select without harming human life. In an article in Medical News Today by Christian Norvitz, amniotic fluid stem cells have many advantages. They are easily obtainable as they double every 36 hours and they may be grown in large quantities. Feeders are not needed to guide them and they do not produce tumors. Specialized cells generated from amniotic fluid stem cells include all three types of cells that exist in developing embryo, and amniotic fluid stem cells have the potential of generating every type of adult stem cell.